And they'll put up these slides, and so you'll be able to find those very recent studies that confirm that same profile. Here's one on the motor system, which talked about when you have skilled motor movements and they become eventually automatic. You know, when you're first learning to type or play the piano, you have to think, where do you put every finger, because where is every key, and what note does that correspond to? But after you are good at it, that becomes automatic, right? You're no longer doing that, and you play rapidly, you play smoothly. So a lot of functions you may learn sort of by thinking about them, but then it becomes automatic. And so this has raised the whole category of automatic processes in the brain. So when you say children with autism don't know the things that all other children automatically know, that's because there is automatic learning. We can call it implicit learning. We can call it categorization. There's a lot of skills like that. And these are skills that are being examined and a significant part of the impairment in autism. The last thing I'll say about fMRI studies is there's never an end to what they're going to find until somebody far in the future gets to the end of the complexity of the brain and the DNA. So the next fMRI finding that presented by our group was, how does the brain think in typical people and how does it think in autism? And to cut to the short version, every word in the brain is represented according to 4 or 5 different attributes. So if you think that the brain has one place that it puts the word for car and one place where it puts the word for house, that's not it. It has a more general descriptive way of characterizing autism, words in autism. The same goes for concepts. Now what that means is typical individuals, if you know that a house is a shelter and you've encoded it partly that way, you can find substitute shelter. You have a flexible understanding of what a house means. But if you have autism, a house is a house, okay? It's not a shelter, it's not, going to help you be flexible. Or if you need a hammer, because we encode in a distributed way, we can grab a shoe and use it for a hammer if we really need to, or anything that has the same shape. So I think once we complete the studies of children, we'll realize that there's too narrow encoding in autism and that we need to try to mimic the more typical way of learning so they have a more flexible understanding of words and ways of doing things. The one thing I don't want to skip is Marcel Just and his colleagues did a study of poor readers, and after 12 weeks of intervention, they could measure changes in the connections, the white matter in the brain. So as Sally Rogers said, behavioral or neurocognitive interventions do have biologic impact. Now, the last thing that I want to say is that when we go to look at all of the genes that have been reported, here we have a neuron with an axon extending out to the neuron next to it, and meanwhile, there's other neurons coming up from the lower part to connect with that neuron's dendrites. Receptive field is the dendrites. Now, genes have ended up being in one of these 3 categories so far. It's either about how the neuron sends its axon out, outgrowth, and then pathfinding. These neurons have to find their connections everywhere precisely in the brain. The second is the synaptic targets. There's genes that interfere with the development and maintenance of synapses, and then other genes that have to do with the dendritic side of the connection. So although we may have a lot of genes, we have them doing common things. And also from child neurology disorders, we know even if you have one gene, there can be a wide range of severity of expression. So that'll give you heterogeneity. But also having different genes will give you heterogeneity. And superimpose that on a family background, and you'll have heterogeneity. Let me see. And this is it. Finding the genes meant that scientists could look at the product of the gene, and that's the protein, and begin to see at a molecular level what was going on in autism, and begin with a biologic intervention, at least in tuberous sclerosis, where most of the children develop intellectual disability, epilepsy, and autism spectrum disorder. They've begun a trial of rapamycin in toddlers with the tuberous sclerosis gene to see if it will prevent the development of ASD and intellectual disability. So that, to me, is one of the beginnings. I think that we will be able to help a great number of verbal children and adults with neurocognitive interventions. And it's important to realize that those can occur at all ages. And when they get older, it becomes secondary socialization, using what they do have for cognitive skills to help them develop a metacognition. If you say this, well, how does it affect that other person? And they can gather quite a lot of improvement from that. And I think the most impaired people with autism in the future will be divided into the category of underdevelopment, axonal pathfinding, axonal outgrowth, dendritic, or synaptogenesis errors, and the gene type, and those will ultimately be treated with biologic interventions. Thank you.